Hey, it's me, Dr. T, and today I want to talk to you a bit about cancer. Cancer is a group of diseases characterized by cells which are growing abnormally, and these cells have the potential to spread and invade the body. They can destroy healthy cells, tissues, and organs. Okay, so how does cancer come about? In order to answer that, I have to give you a bit of a background behind how cells normally grow and what's happening under normal circumstances when things are going right. Your body is made up of trillions of cells, and these cells have different functions. So most cells in your body go through this cycle called, surprise, surprise, the cell cycle. And this cycle is there so one cell can become two cells. So we say one parent cell can be divided into two daughter cells. So in the cell cycle, there are 3 stages, but 2 main stages, which are interphase and mitosis. So this is just so the cell grows and you can produce more cells if needed. So sometimes cells get damaged, sometimes cells are really old and they need replacing. The cell cycle is where this happens. So during the cell cycle, the cell grows, it acquires new materials, and little organs, which we call organelles within a cell, or little structures within the cell, also duplicate. We also have the DNA and some proteins also duplicating within the cell. So mitosis is the part of the cell cycle where the cell actually divides. So during this time, remember I said your DNA is duplicated, and obviously in each cell you want a normal amount of DNA. Remember, your DNA is wrapped up in chromosomes, so a normal amount of chromosomes in each cell. So your DNA equally divides between the 2 cells, and your proteins, and remember the structures I was telling you about, the organelles, also divide equally between the 2 cells. There are so many stages involved in the cell cycle, so there's so many different things that could go wrong within the cell cycle. And so your cell actually has checkpoints. It has proteins that check that things are going well. It has a form of regulation. So if there is damage in any way, usually the cell will be able to repair itself. But if it's not able to repair itself, then signaling happens that kind of ushers the cell towards cell death. It's called apoptosis. The cell kind of kills itself off. In cancer, however, the regulation of the cell cycle goes wrong due to genetic mutations, epigenetic mutations, or chromosomal abnormalities. These are the usual causes of cancer. Remember I said there are genetic mutations that can lead to cancer. Usually, or sometimes, these can be in oncogenes or tumor suppressor genes. An oncogene is any gene which, when mutated, has the potential to cause cancer. So oncogenes start off as proto-oncogenes, and proto-oncogenes encode proteins that are important in cell growth and cell differentiation. So cell differentiation just refers to the production of different types of cells from one type of cell, but you don't need to focus too much on that. The proto-oncogene may acquire an activating mutation, or it may be expressed more than it should be. So if you think about it, if it's expressed more than it should be, if it has this mutation, that means that this gene is continuously expressed, and that means the protein keeps on being produced, and it's kind of ushering the cell towards continuous cell growth, continuous cell differentiation. And so as you can imagine, if the cell is growing when it shouldn't be, this is one of the definitions of cancer. A tumor suppressor gene encodes for tumor suppressor proteins. So basically proteins that kind of stop tumors forming. So these tumor suppressor proteins have loads of roles. Some of the roles include stopping the cell from continuing in the cell cycle if there is some sort of damage. And if the damage cannot be repaired, then it tells the cell basically to kill itself off. We call this apoptosis. These kind of regulatory steps that the tumor suppressor proteins are involved in can help stop the formation of tumors or help stop the formation of cancer. So when a tumor suppressor gene acquires a loss of function mutation, which basically means that the gene loses its function, then you don't get the production of these tumor suppressor proteins, and therefore it can also lead to the development of cancer. I also mentioned epigenetic mutations. So epigenetic kind of refers to changes that happens to the gene that doesn't actually change the information contained within the gene. And these modifications can kind of determine whether the gene is expressed or it's not. So for instance, in the case of DNA repair genes that are involved in repairing your DNA when it's duplicating and things aren't going right, if there are modifications in these genes, epigenetic modifications, you can see that these genes, these DNA repair genes might not be expressed. And so when your cell has damaged DNA, that might not be repaired. And then you can see how the cancer cell is really mutated and doesn't have the right sort of DNA. And that can lead to the development of cancer. So I also mentioned chromosomal abnormalities. Your chromosomes have different genes on them, but sometimes when your cell is dividing, so during mitosis, for instance, the chromosomes might not divide properly, or there may be something that goes wrong when the chromosomes are meant to be, or chromatids are meant to be splitting up into different cells. So for instance, you might have part of the chromosome chopped off, so you lose some of those genes there, or you might have one part of the chromosome chopped off and added to another part of the chromosome. And even in some cases that can form a whole new type of protein which is cancerous or can lead to cancer. A normal healthy cell can become a cancerous cell by a series of events. It might not just have one particular mutation, but usually in one tumor there are loads of mutations. So for instance, the cell acquires the first mutation and then it's not going through the cell cycle properly, maybe it's growing too much, and then it's able to escape other checkpoints, other regulatory checkpoints, other forms of regulation, and then you can see how the cancer develops. There are so many different things that can go wrong in cancer. That's why developing a cure for cancer is very hard, and that's why research has been happening for decades, because there is not just one thing that goes wrong. There are multiple things that can go wrong, even between individuals. Even in one tumor, there can be so many different mutations. But I'm hopeful that as we continue to research into cancer, we should be able to develop treatments that are better than we have now, treatments that are more specific, more targeted towards certain mutations, towards certain type of cancers, certain subtypes of cancers. So thank you for listening. This is me, Dr. T.